So this lecture is on metabolism, and it's a fascinating topic, though I know that sometimes we can find it hard to understand all the different pathways and molecules involved, to the degree that some of us appear to have some sort of metabolism dyslexia. But really understanding metabolism is not half as difficult as it appears, and in this lecture I want to show you how to understand it better, and also to understand all the various processes which are involved in energy production, essentially. Really, the importance of understanding metabolism stems from the comment I made way back in our functional medicine lecture, when I said that the biological science of nutritional therapy is really a nutrient-gene interaction science. And we say that food is medicine, and the new biological sciences are allowing us to understand why food is medicine. And metabolism is an integral part of that understanding. And also to understand the processes behind things like diabetes and obesity, and even cardiovascular disease and cancers and so on, and the energy utilizing processes in the Atkins low carbohydrate diets and sports nutrition. To really properly understand these, then an understanding of metabolism is essential. So by the end of this lecture, I want you to feel more confident in understanding the various pathways and how they link up. And we'll be building on previous understanding, so no doubt before you started the nutritional therapy diploma course, I'm sure you looked at things like cellular respiration and energy production and ATP and glycolysis link reaction and Krebs cycle and so on. So we'll be having a tiny little review of that and building on that quite a bit. So let's have a look at the learning outcomes to start with. So we're going to describe the metabolic pathways by which macronutrients are metabolized and utilized, identify the micronutrients involved as coenzymes and cofactors in these pathways, explain the interrelationship of carbohydrate, lipid or fats and protein metabolism, and explain the hormonal regulation of this interrelationship. So the hormonal relationship essentially, and mostly looking at insulin and glucagon as the two principal hormones involved here. So you remember from your previous studies that all life is comprised of different levels of structural organization. So we start at the atomic level with the little oxygen and hydrogen or whatever type of atoms you care to imagine. Then the simple molecular level with just a few atoms bonded together. Then the complex molecular level. Then we can consider organelles and individual cells and tissues and organs and organ systems, and then the whole organism. So in this case it's a human being. And that's the classic view. But our understanding develops, and the fields of research such as psychoneuroimmunology, which you've had a lecture on, and our understanding of quantum physics and so on, these have really discovered a lot more about life. And we begin to see the inevitable and essentially unavoidable role of the mind or the psyche at the top of the tree of life, and how it influences all the levels below. And while physiologists describe many processes of human biology at the tissue and organ level, and the biochemist studies the same processes but really at a higher magnification. So to a biochemist, the levels below the psyche can all be described in terms of chemical events. So a useful definition of biochemistry is the study of life at the molecular level. And the discipline of cell biology fits between physiology and biochemistry, but the three disciplines together form a continuum of knowledge and investigation. And you remember perhaps back in the functional medicine lecture I spoke about this continuum of health, that we've never got these isolated subunits. It's all joined together. It's true that our current health paradigm considers almost all of the levels of structural organization. So from asking an individual how they feel, so that would be the psyche, or the symptoms they're experiencing in their body, so the organism level, to investigating their biochemistry with a lab test, so that's the molecular level, and drug therapy, pushes molecules around with the intention of getting the cell to behave in a particular way, which will alter the function at the tissue level and the organ level. But there's one common factor to all of these levels, and that's the central role of energy. And energy really is what we're going to be focusing on today. So it's a crucial aspect of where we're going with metabolism and our understanding of metabolism. So what do we study in biochemistry? So biochemical studies follow two key themes. The first one is to study which can be based on the biochemical structures of molecules. So for example, glycogen or DNA or protein conformation, so how the protein is formed and fits together, or the structural interrelationship between the molecules. So for example, enzymes with their substrates or hormones with their receptors. And the other theme in our study of biochemistry considers the numerous dynamic events known collectively as metabolism. And metabolism can be defined as the sum of all the chemical changes occurring in an organism. It provides the energy and the building materials required to sustain life. And this lecture will be focusing on just that. So metabolism, the word from metabole, meaning change. And metabolism, as you perhaps know already, is split into two different subjects and subsections. We start with catabolism, and that's the degradative reactions, so the degrading reactions, including the oxidative breakdown of nutrients to yield energy. And catabolism also creates products which can then be used in anabolism. And anabolism are those biosynthetic reactions, including those required for growth and energy storage, and using smaller molecules to make larger molecules, and so on. So we have these catabolic pathways. For example, we ingest food, we digest food, we have to break the food down, and the products which have been broken down from the food can then be used in these anabolic pathways to create new molecules, and so on. So all metabolic processes undergo a conversion from one biochemical substance to another, and this may be one step or many steps. And typically enzymes are the workhorses which actually make the conversion, and the resulting product will normally then go on to be metabolized into another product that can be used in the body. And these building blocks are called substrates, and the substrate is defined as a molecule upon which an enzyme acts. Enzymes catalyze biological reactions involving the substrate, or the substrates, and the substrate binds with the enzyme's active site. You remember this, I'm sure, from your previous studies. And the substrate is then enzymatically transformed into one or more products, which are then released from the active site. So that's just a little review. Now, looking at what we're now calling the metabolome. Now, you remember in your genomics lecture, we were talking about genome and maybe the transcriptome and the microbiome to discuss all the different variable bacteria which all connect up together and have a role to play inside our bodies in the world, and so on. And the metabolome is using the same sort of concept, refers to the complete set of these small molecule metabolites such as metabolic intermediates, hormones and other signaling molecules, for example, and these secondary metabolites as well. And they can be found within a single organism. So we talk about the metabolome of the human body, or the metabolome of a rat, or metabolome of any other sort of organism, be it a plant, animal, or anything, any other life organism which undergoes metabolic reactions, which is every life, or all life. The word metabolome was coined in analogy with transcriptomics and proteomics. And like the transcriptome, so the complete set of all the transcriptional molecules, and the proteome, so the complete set of all the different proteins which we have in the body, the metabolome is dynamic, and it changes from second to second. So let's have a look at catabolic processes. And here we see catabolic processes are when energy is being released, and that's the simplest definition of what a catabolic process is. The root word catabolic, I think, is the same as that as catastrophe. So this breaking down process. And we'll be looking at all these different things during the lecture. But let's just have a little look through. So we're starting with glycogenolysis. And lysis refers to the splitting. So lyse, L-Y-S-E. And to lyse means to split or to break apart. So you can be assured that any process with the word lyse or lysis in it will be a catabolic process. And when you lyse glycogen, what do you get? So remember, glycogen is the storage form of glucose. So when you lyse glycogen, you get glucose. Then we have glycolysis, so the splitting of glucose. The Krebs cycle is the final breakdown of carbon dioxide, so CO2, the final breakdown to carbon dioxide from acetyl coenzyme A. So that's a catabolic process. Then we have lipolysis, so lipolysis, splitting of triglycerides, not the breakdown of fatty acids, which is often a common mistake people make. So lipolysis is the splitting of triglycerides, and that's into glycerol and the 3 fatty acids which it's comprised of. The oxidation of fatty acids, the deamination of amino acids. We'll be talking a bit more about what deamination is a bit later on. And then the end products of these processes include things like carbon dioxide, water and ammonia. So those are catabolic processes. Now we have anabolic processes. And in anabolic processes, when you're building up new molecules, I'm sure it's quite obvious that energy is required in this process. So let's have a look through these. So protein synthesis, creating new proteins and nucleic acids as well. It's obviously a building process. Storage of glucose as glycogen. So basically we are compacting glucose molecules together, and so that's a building up process, and then we're storing them. So the storage form of glucose is glycogen, and it's stored in different tissues around the body, and we'll talk a bit more about that later on. Then the conversion of glucose to fatty acids and triglycerides. So glucose, as I'm sure you remember, is a very small molecule, and then fatty acids are a little bit more complex, and triglycerides are more complex again. So as we're building up complexity, this is naturally going to be an anabolic process. Then cholesterol and steroid synthesis. So the word synthesis here is a bit of a giveaway. So we're creating something. So that's going to be an anabolic building process. And also phospholipids and proteoglycan synthesis. And they're used as structural molecules in the body. So anabolism is the creation of new molecules that the body uses, proteins, nucleic acids and so on, and stored energy in the form of glycogen and fatty acids. Now then, perhaps some of you have seen this poster in the polling room at ION, and it's a bit of a daunting one sometimes, but really what you'll probably find is, hopefully, when we get to the end of this lecture, you'll have a bit more of an understanding about how this all fits together, but perhaps more importantly, you'll be less daunted by it, or less threatened by it. It is estimated there are between 2,000 to 3,000 different types of metabolic reaction occurring within human cells, and just so you know, there are probably no biochemists in the world who know all of these metabolic reactions, certainly not off the top of their head. Some are common to all cell types, these reactions, while others are restricted to just 1 or 2 particular tissues, whose specialized physiological functions reflect the specialized metabolic changes occurring within them. And this poster essentially represents a large number of these metabolic processes, but not just metabolic processes, we also call them metabolic pathways. And as perhaps you can see, they are all, one follows the other, and one feeds into the other. So it's the substrates of one reaction which is going to become the substrate of another reaction, and so on and so on. Metabolism is fascinating, but at first sight you can't deny that it does appear quite complicated, and I'm sure you feel quite daunted by the challenge. But for most students, it's unnecessary to learn every reaction in every pathway. What's important is that there's an understanding of concepts of metabolism. So what appears to be a complicated set of reactions, like we see here in front of us, and pathways, can be seen in terms of relatively few chemical and thermodynamic principles. And we're just going to go on and find out how best we might be able to understand that. So let's have a look at the journey of metabolism. Metabolism can be likened to a journey. There's a starting place and a destination, and there will be some important intermediate stops, perhaps where a change of mode of transport might be necessary. There'll be points of interest and also places en route which have little or none of our attention. And it's really vital to realise that metabolism is adaptable. So changes in our physiological situations, so whether we're in the fed or fasting state, so whether we've recently had a meal or we've not been eating for a few hours or even a few days, resting or exercising, health or disease, and these all result in changes in particular aspects of our metabolism. So metabolic pathways. So let's just start to have a little look at metabolic pathways, try and develop our understanding of what they're all about. Metabolic processes occur via a series of individual biochemical reactions. Such chemical reactions are usually under the control of enzymes, which I'm sure you know, which act upon a substrate molecule and produce a product molecule as shown in this diagram. So here we go, there's a substrate, there's the enzyme, and then there's a product. So pretty simple. The substrates and the products are referred to collectively as intermediates or metabolites, and the product of one reaction becomes the substrate for another reaction, and so the concept of a metabolic pathway is created. So here we go, it's a simple pathway. So we've got substrate A and then enzyme A. Perhaps you can remember that most enzymes, or anything ending with ASE, such as lipase for example, is an enzyme, and that catalyzes the reaction from substrate A, and that becomes substrate B. And then so the second enzyme then catalyzes a reaction from substrate B to substrate C, and so on and so on. There we go. So the conceptual module is simply a way of trying to explain experimental data. A potential problem in representing metabolic pathways, as in this slide, is that there's an implication that they are physically or topographically organized sequences, but in reality this is not necessarily true. Although there's no doubt that this concept helps us to understand metabolism, particularly in the early days of our learning. So it's a really useful concept to be able to get hold of, just put in your mind and be happy that this is essentially what's going on within the cell. So with some exceptions, most enzymes are found free within the cytosol, so like in the top left-hand corner of the diagram, or rather even in a compartment of the cell, where reactions occur when an enzyme and its substrate meet as a result of their own random motion. So sort of in the so point B in the middle of the cell there. Clearly this would be very inefficient were it not for the fact that cells contain many copies of each enzyme and many molecules of each type of substrate. Because I know often it's a bit confusing to think, do they just randomly meet up? At the moment our understanding is yes, but we also understand that there's quite a high concentration of each of the enzymes and the substrates, so it ensures that they meet together and are able to perform their different reactions. So the importance of enzymes is demonstrated here in this diagram. This diagram's from the Laboratory Evaluations book. Really, of the myriad of different processes which affect nutritional status, they are central to digestion, as you can see in the second boxed along digestion of nutrients, enzymes form a central role. But that's not to forget that enzymes are not only in the digestive system but all over the body in all the cells. It is a common misconception that nutritional therapists sometimes think that enzymatic metabolism often occurs outside the cell, because the enzymes they're most familiar with are the supplemental digestive enzymes, which are indeed released into the lumen of the small intestine. And there are also enzymes released by the pancreas into the small intestine, but there are also brush border membrane-bound enzymes. So remember the brush border of your small intestine, the villi and the microvilli. And within the body, where most enzymes are found, it's within the cells that they reside. So I just wanted to give you an example of enzymes in action. And this diagram, which I'm going to build up, is going to show us how dietary protein becomes the neurotransmitter and hormone dopamine. So we ingest food, we digest the protein in the food, we absorb it into the body, cross the brush border, and then that's metabolized into phenylalanine. Now that process requires hydrochloric acid to break down the proteins in the stomach, an enzyme called carbonic anhydrase, which is zinc dependent, and then also proteases, which are enzymes which also break down protein. So phenylalanine is then metabolized into tyrosine, and that requires phenylalanine hydroxylase, so that ends in ASE, so obviously that's going to be an enzyme. And then we have another enzyme, tyrosine hydroxylase, which is iron and tetrahydrobiopterin dependent, and that creates the product levodopa, or L-dopa. And then we have dopa decarboxylase, another enzyme again, of course, which is vitamin B6 dependent, and that is synthesized into dopamine. However, tyrosine, if we go a couple of steps back again, is also metabolized into thyroxine, which is the hormone produced in the thyroid gland in the front of your neck. And that requires deiodinase, which is iodine, selenium and iron dependent. So really, depending on your physiological or biochemical requirements, tyrosine can be metabolized into either L-dopa or thyroxine. So here we're just building up this metabolic pathway and showing you how one thing may be converted into another and what is required as a result. But let's go back to dopamine, because dopamine itself can be then metabolized into noradrenaline, which is dopamine beta hydroxylase dependent, using copper and vitamin C. Then noradrenaline can be metabolized into adrenaline, which uses methyltransferase, which S-adenosyl methionine is used in that process. And then adrenaline can be metabolized into cortisol and DHEA, which, as you probably know, are your long-term stress hormones. So cortisol is itself metabolized from DHEA, and that uses vitamin C in that process. So here you can see lots of different substrates, lots of different enzymes, and the required essential nutrients which are involved in that process. What's interesting is that those nutrients are often not something which other health professionals are aware of. They know about these processes with regards to enzymes and the substrates, but they don't know about the critical use of the micronutrients involved in those processes. So this is something which your expertise is going to be able to be utilised with regards to your patients or your clients, and then you'll be able to help them much more effectively. Now, let's move on. Metabolic map reading skills. The reason I put this up here is to show you that really this metabolism pathway is like a big map. And you remember the metabolic pathways poster or map which I showed you earlier. Think again about making that journey. A useful analogy is a road map of a city centre, where there are these main and subsidiary roads, one-way systems and junctions. where traffic flow is controlled by signals and roadside signs and so on. And very few people would attempt to learn or memorize the complete road map, unless you're a taxi driver perhaps, a black cab driver. But learning the rules of the road, coupled with basic map reading skills and the knowledge of the main roads, will enable most people to successfully negotiate a journey from one place to another. And a complete diagram of intermediary metabolism appears to be as complicated as a road map or of a city or a region. And it's just a big tangle of individual reactions within numerous substrates. But understanding biochemical pathways is really very, very similar to map reading. And the flow of traffic along the roads and through the city is conceptually similar to the flow of substrates within the cell. And rather than visualizing cars and vans and buses, think about the numerous carbon and hydrogen and nitrogen, phosphorus and oxygen atoms flowing as component parts of substrate molecules and through the pathways within the cell. And just as the traffic flow is regulated and directed with signals and restrictions, so too is the flow of substrates. So we can say the vehicles, or the metabolites, if we're going to call them that, they join or leave a particular traffic flow at interactions, converging or diverging pathways, for example, and the rate of flow is affected by traffic signals, which we're going to call enzymes, by roadworks or accidents, which we're going to call defective enzymes, and by the number of vehicles using the road, which we're going to call the concentration of the substrate molecules. And they may need to take shortcuts or be diverted to avoid congested areas, and the body can do that as well. So similarly, substrate molecules also may be routed via alternative pathways in a manner which is, best serves the physiological requirements of the cell at any particular moment. Okay, so strategy for learning a pathway. Active learning is essential. When you're asked to learn a pathway, the temptation is to sit down and try and memorize each step in turn from top to bottom. So this, as perhaps you're familiar with, is glycolysis. A common failing in students who are new to metabolic biochemistry is in trying to memorize the whole of a pathway at the outset. So, rule number 1, resist the temptation to memorize. This approach leads to knowing but really not understanding. And metabolism is a real mosaic of component parts. And just as we nutritional therapists know that nutrients do not work in isolation, so performing experiments on single nutrients has little value, so metabolic pathways don't exist in isolation either. In fact, nothing in life does. So start with a lower power view. Try to see the pathway in relation to others and be clear about the physiological purpose of the pathway. Taking the time to see the broad picture at the start of the learning process will make the learning process more meaningful and therefore quite a bit easier as well. And after all, that is what we're seeking, I think, isn't it? We're trying to find meaning in our learning. So that's rule number 1. Rule number 2, be positive. Do think about the pathways as more than simply information gathered from experiments carried out in test tubes. Do think about the biochemistry as infinitely more than a body of knowledge that has to be mastered to pass an exam. Think about what's going on inside your own cells and tissues at various times. Just look down at your hand and think about all the myriad, thousands and thousands of different biochemical processes which are going on inside all of your cells at every moment. And then, having attended a lecture or gone for a walk in nature, think about how reactions and pathways in your own cells and tissues respond to your changing physiological circumstances, such as sleeping and sitting and walking and running fasting after food. All of these different processes have an effect on your biochemistry, and we'll find out a bit more about that later on. So biochemistry is this wonderful dynamic study, and it's really all about you. So let's have a look at a strategy which we might be able to employ to help us learn actively about how you put a pathway into its proper context. So number one, where does the pathway occur? That is, in which cell types or tissues, which compartment of the cell? Is it the cytosol, the mitochondria, the lysosomes, etc.? What is the biochemical purpose of the pathway? So, for example, to release energy, or to produce a key functional molecule, or to synthesize a macromolecule, perhaps? What are the links there are between the pathway and other pathways? Where are the control points between the pathway? And when does the pathway operate? So is it always active, or is it an adaptive pathway? So let's have a look at an example. So put that into practice. So we're going to be talking about glycolysis here, as you can see at the top. So it's universal. It occurs in all cell types, and it's cytosolic. So it's performed in the cytosol. So what's the purpose? To begin the oxidative catabolism of glucose. So the production of ATP. Small production. So this is not a prime role in most tissues. So do you remember glycolysis being the beginning of this cellular respiration process? We only get 2 molecules of ATP. You may remember this from a previous study from this process. Then the end products of pyruvate or lactate, if you're overdoing it a bit, are important compounds for other pathways. What are the links to other pathways? So they are the pentose phosphate pathways and glycogen metabolism. We'll be having a bit more of a look at these later on, so we'll help you understand that a bit more as we progress. And glycerol from lipids may enter at the level of triose phosphate. So where are the control points? So you may remember these enzymes. Don't worry about it if you don't. There are different enzymes: hexokinase, glucokinase, phosphofructokinase, and pyruvate kinase, which are all involved in this process of glycolysis. When does the pathway operate? All the time. So let's have a look at ATP and ADP. I'm sure you're familiar with ATP, the currency of energy in all living organisms. It's a small water molecule, a small water-soluble molecule, so it's easily transported around the water-rich environment of the body. And in healthy individuals, it's very rapidly synthesized and used up, and there's a rapid turnover of ATP in the cell. So we start with ADP, so we've got adenosine and ribose and those 2 phosphate molecules. Then that is synthesized by a phosphorylation reaction into ATP, and we've added the 3rd phosphate molecule. And there on the left is just sort of a cyclical diagram, just to remind you how ATP, the energy from the 2nd and 3rd phosphate bonds, is used up, and so that becomes ADP again, and then that's resynthesized through that phosphorylation reaction to become ATP again. And the process goes on and on and on. In the human body at any one time, there's about 5 grams of ATP. But we have to eat all the time, we have energy stores in our fat and glycolysis and so on. So in any one day, you'll use about 40 kilos of it. So basically, to get from 5 grams to 40 kilos, this is telling us that it's very rapidly recycled all the time. And we are constantly feeding in more energy to resynthesize that ADP into ATP as a continuous process. So cellular energy production, or cellular respiration as it's also known, the universal currency of energy in living organisms, used in all forms of life. So every organism, be it a plant, bacteria, a mammal, anything you can think of which is defined as having life in it, uses energy, cellular respiration. There's a small water-soluble molecule, mentioned that, transported easily around the cell and between cells. It's got this rapid synthesis and breaking of those high energy bonds, many times per second, and allows energy to be generated and used very rapidly, ensuring a constant supply. So you remember back in the functional medicine lecture again, we had this continuum of health and disease. And really this, I'm just putting this in again, just to give you an example of why ATP is so very important. One of the major health problems you'll encounter as a nutritional therapist is a lack of energy. And by this I don't mean thinking about the superficial outer presentation of simply just feeling tired. A lack of energy permeates much deeper and directly relates to how efficiently the individual cells and the mitochondria are synthesizing and using energy. And as a practitioner, one of the very first questions I ask my clients is, how is your energy? Because really it's the ultimate telltale sign of whether there's a problem of cellular dysfunction. So remember, we have illness right down the bottom, and then that's characterized by disease and symptoms and so on. And we've got sort of intermediate health where you may have a few symptoms, and then we have optimal wellness, which is beyond what we may call health. So if you consider that a lack of energy might be around the health area, we've already started the pathway over to the right to down the disease process. So we say that energy is this telltale sign of a potential problem with cellular dysfunction. If your energy is low, particularly on a chronic basis, there's a problem. So in a way, a good mantra to learn is a chronic lack of energy is a sign of early pathology. So a chronic lack of energy is a sign of early pathology. And there are many health professionals, particularly interestingly American cardiologists, so consultants who specialize in heart health, they are becoming very aware of this lack of energy and how it's such a crucial part of cellular function, and how a chronic lack of energy can be a clear sign of early stage pathology. Certainly people with advanced stages of disease typically have low energy levels as well. So where does our energy come from? That provides the energy in a utilizable form. The energy is normally stored in the bonds of molecules that are in the macronutrients from our diet, so the fats and proteins and carbohydrates. And we break these down, break the bonds, oxidize the compounds, release the energy, and that energy is transferred to ATP by those cellular respiration processes. And because we learn cellular respiration and carbohydrate is the usual substrate, we often think that carbohydrates are the only source of energy. While it's the cell's preferred source of energy, certainly, remember that the body can also use all of the macronutrients for energy, so amino acids and triglycerides as well. And they can all feed into the cellular respiration at different points. So phosphorylation. We mentioned phosphorylation. That's the synthesis of ADP to ATP. It's a phosphorylation reaction. So it's typically kinase enzymes, Kinase, kinase enzymes, which perform these phosphorylation reactions. So they're an important family of enzymes. Because it's called phosphorylation because it's adding a phosphate group. I'm sure you know that. And there are two different ways to synthesize ATP. We've got the substrate level, It's a simple transfer of phosphate from the substrate in glycolysis or the electron transport chain to ADP to make ATP. Then we've got the oxidative or electron level phosphorylation, and that happens after the Krebs cycle in the electron transport chain, which I'm sure you remember. So catabolism of carbohydrates, lipids and proteins as fuel. This slide is sort of the essence of this lecture, so let's go through it each line at a time. So all of these three macromolecules can be catabolized or broken down to yield energy. Carbohydrate in the form of glucose, so when we eat carbohydrate food we break it down into the simple glucose molecules. As a principal energy source for all cells, can use glucose for respiration. Fatty acids are also very important. Many tissues use fatty acids as a major fuel, and we'll explain a bit more about that later on. And then proteins, or their constituent amino acids, principally used for structural and functional molecules, but can be deaminated, and again I'll explain that later on, and converted to carbohydrate metabolites to be used as fuel. So let's have a look at this overview of macronutrient metabolism. And this diagram here on this slide is actually from chapter 7 in your textbook, the Gropper Advanced Nutrition and Human Metabolism book, so you can go and have a look at that and read all about it. I highly recommend you do that. And this diagram shows the interrelationship of the three macronutrients. You can see at the top protein, carbohydrates and fats, or lipids, and how they each feed into metabolism. So we can see at the top the different macronutrients. You'll remember from your previous study that carbohydrate is the most commonly used source of energy in the form of glucose, which passes through glycolysis, the link reaction, Krebs cycle, electron transport chain, and oxidative phosphorylation, and so on. But also we can see that fatty acids and glycerol, which combined form triglycerides, they feed into glycolysis as well, and then into the Krebs cycle to generate ATP. And also protein, which is broken down into amino acids over on the left there. So protein, amino acids, methionine, tryptophan, phenylalanine, tyrosine, and so on. All of these different intermediates, these metabolic intermediates, they can be broken down. These amino acids first have to be deaminated. So if you enzymatically remove the amino group from an amino acid, you essentially have a carbohydrate molecule which can be fed into carbohydrate metabolism. Again, I'll be explaining a bit more about this later on to give you a clearer idea about what that's all about. But because you'll know that you have different types of amino acids, I'm sure you've learnt this from your studies and your essay, they generate lots of different compounds which feeds into carbohydrate metabolism at these different points, as you can see. So the reason they're coming in at different points is because of those different types of amino acids which we have. Some can be converted into pyruvate. So you see pyruvate in the red box in the middle of the diagram there. Others into acetyl-CoA, just to the right of the pyruvate there. Others feed into the Krebs cycle directly, as you can see a bit lower down. So that's just an overview of how the macronutrients are interrelated in their roles of energy synthesis, but we'll see more clearly how all this works later on. Now I mentioned how we've got these micronutrients which are very important in these processes, and there are quite a few of them involved, particularly B vitamins, which are critical for energy production in these catabolic processes and cellular respiration, and they're required as coenzymes for many of the reactions to occur in these pathways. They're organic, so that means they're these carbon-based molecules, but are much smaller than the enzymes they assist, and typically involved in a certain type of reaction, but in many different places. So here's a summary of the different B vitamins, which are involved in these different types of processes and the different coenzymes which they are assisting. So, looking at the chart, vitamin B3, niacin, for example, is a major component of NAD, which is the electron carrier, which functions between glycolysis and the Krebs cycle. You might remember that from your previous studies, and carries electrons from those processes to the electron transport chain, and is also involved in oxidation and reduction. So, remember oxidation and reduction? So, redox reactions, as they're known. Pantothenic acid is the main component of coenzyme A, which attaches to the acetyl group and feeds into the Krebs cycle. So B vitamins play an essential role in this energy production as coenzymes. And you remember that these vitamins are being used inside your cells to produce energy right now. So you're sitting listening to me, and your heart's beating, which requires energy from these processes, and your lungs are breathing thanks to your diaphragm raising up and lowering, and your brain cells are functioning and able to think thanks to the energy from these processes. So without these vitamins and nutrients, none of this would happen, and all your cells would literally just stop working. So glucose metabolism. If we have a look at an overview of glucose metabolism, you can see the dotted horizontal line represents the boundary between the cytosol, which is the top part of the diagram, and below in the mitochondrion. And I'm sure that you're familiar with this diagram being that of cellular respiration. But this diagram also shows you in yellow some of the critical micronutrients used in these processes: magnesium, manganese, the B vitamins, and alpha lipoic acid also. But of course, glucose comes from carbohydrates, and this might be a bit of healthy revision for you after your macronutrients lecture. Remember that dietary carbohydrates are digested and absorbed as monosaccharides, so those on the far left of the diagram. So the monosaccharides are glucose, fructose, and galactose, and these combine to form the disaccharides, which are two monosaccharides joined together. So the disaccharides being the next group of simple carbohydrates just along to the right. And you can see that both mono and disaccharides are classed as simple carbohydrates, whereas if you start stringing more sugar molecules together, being 3 or more, so the oligosaccharides, you get into the realms of complex carbohydrates. And it's when we have more complex carbohydrates bonded together that the body has to work that little bit harder to break these bonds apart so the sugar molecules can be absorbed from the gut. And that process takes a bit longer, which results in the sugars being released more slowly. So eating the complex carbohydrates has a much more gentle effect on the body. And of course there are some carbohydrates made exclusively by plants, of which their bonds can't be broken in the human body at all, because we lack the enzymes to do so. Although other animals, such as ruminant animals like cows and sheep, they do have the genes to synthesize these enzymes. So in humans, their structure remains undigested. And of course these carbohydrates are classed as dietary fibre. So over on the far right there. I said these are made exclusively by plants, which means that animal products, so meat and fish and dairy, etc., contain no dietary fibre at all. And dietary fibre, because it can't be digested, acts as the scouring pad of the digestive system, shall we say, and also binds toxins and excreted hormones and other waste products in the gut to be removed out in the stool. So the destination of dietary carbohydrates. And you'll remember from your digestion lecture that carbohydrates are absorbed through the intestinal wall, mostly as glucose, and carried via the hepatic portal vein, straight to the liver for checking over. Remember the Kupffer cells, part of our immune system. They sample whatever passes through, and those carbohydrates are then mostly metabolized. Once the glucose has passed through the liver, it enters the systemic circulation around the body and is carried into our cells via the glucose transporter proteins. And we'll have a bit of a closer look at those later on. We've also got galactose and fructose taken up by the liver and converted to glucose derivatives. We'll be looking at that a bit later on. Glucose, the most abundant, extensively metabolized by the liver, but the remainder passes into the circulation and it's transported, dissolved in blood plasma, enters the cells via these glucose transporter membrane proteins. And again, we'll be having a bit of a look at those right now. So glucose transporter proteins. We used to think there are 7 glucose transporters, and indeed the old textbooks will obviously still say this, but currently we know that there are now 12 transporters of glucose. And these are molecules which sit on the edge of every cell to allow glucose to pass through. Remember, glucose is a water-soluble molecule. It can't easily slip through the phospholipid bilayer, this fatty bilayer of the cell membrane, so needs to be carried across by a transporter, which is essentially a little channel created in the cell membrane, especially for glucose. And this is where glucose transporters come in, and they're abbreviated to GLUT, or GLUTs, and these are what's known as transmembrane proteins. So they span the phospholipid bilayer of the cell membrane to carry in glucose used for energy. And there are different types of glucose transporters for different cells, as you can see on this table here, and that's again from the Advanced Nutrition and Human Metabolism book, so you can look that up and read about it. But you can see it's only the GLUT4 which is controlled by the hormone insulin, and that's quite an important thing. Let's just have a look at a diagram of the glucose transporters. This diagram shows you what happens, and it's a great example of the very dynamic nature of cellular function, I think. So when insulin levels are low, we have these little vesicles shown as circles on the left of the diagram inside the cell, and they remain within the cell with the GLUT4 proteins on their little membranes. So the little pink splodges, they're the GLUT4 proteins. When insulin binds to an insulin receptor on a cell at the top of the diagram, could be an adipose cell or a muscle cell, that triggers a cascade of reactions. Don't worry about having to learn anything about that cascade. All you need to know is that this results in these GLUT4 vesicles merging with the cell membrane, as you can see them doing over on the right hand side of the diagram. So the GLUT4 proteins then become an active part of the cell membrane and are able to carry across lots more glucose into the cell than would be otherwise, and the glucose being the little green dots. So this is all triggered by the insulin, which binds to the insulin receptor on those cells. So utilization of glucose. What happens when the glucose gets into the cell? Well, we know it's used by all the cells to produce energy. Central nervous system and erythrocytes, so the red blood cells, only typically use glucose, particularly the red blood cells, under normal conditions. Any excess glucose is stored as glycogen in your hepatocytes, so your liver cells, and muscle tissue as well. And any further excess of glycogen, any further excess of glucose is then metabolized into fat as triglycerides, and then ferried off to be stored in your adipocytes, so your fat cells. So once in the body, glucose is either used or stored as glycogen or as fat, and I'll explain a bit more about that later on. I just wanted to show you an overview of aerobic respiration in terms of the speed that energy is released. And remember that aerobic respiration is a series of enzyme control reactions, so glycolysis link reaction, Krebs cycle, and so on. And that release, they release the energy stored up in carbohydrates and lipids and amino acids as well if necessary. The importance of the control of the enzymes becomes apparent when we realise the effect that releasing all the energy from a meal in one go would have on the body, and this would not be a very pretty thing. So we see that burning all the glucose from carbohydrate, for example, let's start with food, we digest the food, we release the energy, and there's C6H12O6, which is the chemical formula for glucose. If we released all the energy in one go, and it would be oxidized to carbon dioxide and water, this would be quite a lot of energy being released. So the single step oxidation releases all the energy in one go. But this would be hugely problematic for the cells, simply because it would just be too much energy to cope with in one go. So one of the things we're going through, one of the things we're doing through the metabolic pathway of cellular respiration is gradually controlling the release of energy step by step. There we go. So that energy can be harnessed efficiently as ATP. Okay, so the first step is of course glycolysis, then the link reaction, Krebs cycle, and finally oxidative phosphorylation and the electron transport chain. So there's 4 steps of gradual energy release. And just to illustrate this point, I found quite a useful short video demonstrating what would happen if all the energy from a simple meal was released at once. And this is from the BBC series Bang Goes the Theory, and hopefully it'll give you an idea about what would happen if you ate a little simple meal of carbohydrates and all the energy was released at one go.
As human beings, we get the energy out of our food by reacting it with the oxygen in the air we breathe. But that takes quite a long time and we can't see it. So Sydney here has agreed to mix our flakes directly with oxygen to speed up the process a bit. Now, this has never been done before, so I'm more than a little intrigued as to the result.
Remember, Sydney's using nothing except breakfast cereal, oxygen and a little detonator to start the reaction.
Firing! 4, 3, 2, 1.
The energy that you see in that explosion is exactly the same energy that you produce in your body chemically from a packet of these. And to know there's that going off in my body between breakfast and lunch is almost concerning. I might cut down.
Okay, so that was quite a dramatic example of what would happen if all the energy from a small carbohydrate meal was released all in one go. So I'm sure you can see now the importance of being able to gradually release this energy throughout the body in this stepwise process. So we've sort of touched on glucose there. It's quite a common sugar, but I want to talk about fructose now, because fructose is the sugar that's been receiving increased attention in the scientific research recently. It's a sneaky fellow and unique in many ways. Ordinarily, glucose is absorbed directly from your intestines and enters the bloodstream. If you eat a significant amount of sugar and your pancreas secretes insulin, a growth hormone, that this anabolic hormone that tells your body to store sugar as fat into your bloodstream. And as insulin levels rise as a result of glucose ingestion, this feedback system in the satiety center, so the satisfaction center that you've eaten enough in your brain, gets activated. And additionally, the presence of glucose in your bloodstream releases a fat regulating hormone called leptin, L-E-P-T-I-N. And leptin signals your brain that you've had enough to eat. So glucose transported in your bloodstream gives your brain a double dose of hormones telling you to stop eating. Now, how does a plant override in animals the I'm full switch? What it does, it produces fructose. And the reason it wants to do that, if we're talking evolutionarily speaking, is because when we eat plants, particularly fruits, they carry the seeds. And if we are going to be eating a fruit, we usually, or mammals normally eat the seeds as well. They'll eat the whole fruit, like an apple or something like that. And then they'll eat up all the fructose and all the carbohydrate, which is the primary macronutrient of the fruit. And then they will basically, in the stool, will be carried the seeds, which have been protected because they've got these hard little coatings to them, these hard shells, which the seeds are carried in. And they're basically dropped on the ground with a nice dose of fertilizer, being the stool, and then they can grow dispersed away from the parent plant, so they're not in any competition with the parent plant, to grow into a nice new plant. So plants need to override that I'm full switch so the animal can eat more of the fruit and thus disperse more of the seeds. What it does, it produces fructose, which is another form of sugar, which circumvents the activation of both these warning signals. So rather than enter the bloodstream, fructose goes directly to the liver, where, among other things, it stimulates the formation of triglycerides, the precursor of cholesterol. Now, remember about triglycerides, because I'm going to talk a bit more about these later, and why fructose is now perhaps not being seen as such a great sugar to be consuming after all. Because fructose doesn't enter the bloodstream, insulin rises much more slowly. So it goes straight to the liver, remember. So insulin is not triggered, and leptin certainly isn't activated either. So signals, remember, leptin signals your brain that you've had enough to eat. So sneaky fructose bypasses both the feedback defense mechanisms your body has developed to overeating. And you may have read about the problems American cousins are having with high fructose corn syrup, which is an increasingly common food additive, which has been receiving a lot of attention in the nutrition and medical literature recently. And in the UK, high fructose corn syrup is called glucose fructose syrup. So have a look out for that on your processed food packages and the ingredients list. And this metabolic process I've just described, which is one of the concerns about its effects on the body. So this bypassing of those signals. Okay, so I just wanted to highlight fructose with regards to glycolysis. So remember the first stage of cellular respiration. And what's interesting about the glycolysis pathway is that the enzyme phosphofructokinase, which you can see as the catalyzes the third step, is the slowest enzyme in glycolysis. And in biochemistry, this is called the rate-limiting step of the reaction. Literally because it slows the rate of the reaction down. So we have glucose starting at the beginning of glycolysis over on the far left, but when fructose is catabolized in cellular respiration, it can Q-jump, and it can feed directly there in a bit further down, and is converted into fructose 1,6-diphosphate, or biphosphate. And it's not hindered by the rate-limiting phosphofructokinase. So when you catabolize fructose, it's faster than the glucose because you skip past the rate-limiting enzyme. And because a metabolic pathway is essentially only as fast as its slowest reaction, when fructose is oxidized, it goes through glycolysis very, very quickly in relation to glucose. But the caveat here is that fructose is not metabolized in all tissues, mainly in the liver and the kidneys as well, and small intestine epithelial cells, I think, also can. They also have the enzyme to convert fructose into fructose 1,6-diphosphate and jump the Q in glycolysis. Whether or not this is a factor in the research linking these high fructose diets to metabolic diseases such as type 2 diabetes or obesity is still open to question. But then fructose is still recommended to people with diabetes, principally because it doesn't stimulate the release of insulin from the pancreatic beta cells, which I'm sure you know, diabetes. People with diabetes have very low levels of that. And then let's not forget about the other absorbable monosaccharide, galactose. One of the sources of galactose is the disaccharide lactose, which is a polymer, so more than one molecule, of a molecule of galactose and a molecule of glucose. So lactose is galactose and glucose bonded together. Lactose is catabolized by the enzymes lactase, and galactose can either be converted to glycogen, which is the storage molecule, or to glucose as well, as you can see down the bottom of that diagram. There we go. May also be used to synthesize glycolipids, glycoproteins, and glycosaminoglycans. And they are all essentially three structural molecules in the body. So let's have a quick look at mitochondrial dysfunction. If there are problems with the mitochondria and they're not functioning efficiently, as is the case in many diseases, as we mentioned before, then you're going to be more reliant on glycolysis, which not only doesn't produce as much ATP, far from it, as the remainder of the cellular respiration pathways, but as also kicks off its own waste products such as lactic acid. And both of these factors can contribute to health conditions associated with mitochondrial dysfunction. So fatigue is the most obvious result of mitochondrial dysfunction, but there are also other physiological systems that can either be sensitive to increased metabolic waste, such as the nervous system, so neuropathies, so neuro being the nervous system, pathy being pathological. So the metabolic waste of the nervous system or insufficient energy. And we tend to think of the heart as being very energy hungry, and indeed it is, because it's a very visible user of energy, needing to continuously mechanically pump blood for an individual's lifetime. But other organs also use a huge amount of energy. So the liver and the kidneys, for example. Just because it doesn't move like the heart, it still requires a lot of energy to function, which is why, as a nutritional therapist, I would consider liver function in someone whose calorie and nutrient intake is quite low. And mitochondrial function is also associated with aging, accelerated aging, and the diseases of aging as well. So let's have a look at nutrients for mitochondrial function. As I'm sure you can guess, the B vitamins are important. Antioxidants, glutathione, which your body synthesizes itself, vitamin C and E, which gets from the diet, carotenoids, there's carotenes, flavonoids, these plant phytochemicals, all essential. Coenzyme Q10, I'm sure you remember that as the critical, one of those 4 critical steps in the electron transport chain. Carnitine, lipoic acid, and organic acids, which are intermediaries from the Krebs cycle. So as well as considering the functional medicine matrix for the antecedents, triggers and mediators of mitochondrial dysfunction, these key nutrients could all be considered, which all play a role in the synthesis of energy.